I've collaborated with JGI, I want to say it's probably been about 10 years, just kind of off and on. We've been doing these one-offs for high molecular weight DNA for a few projects, and then the ETOP program came around and we applied and actually are now providing high molecular weight substrates for whole genome sequencing as well as for genotyping of various crop plants, various flagship genomes for JGI, and also in support of the community science projects. The reason why this is this project is so important is that many labs in the world, they can isolate DNA, but it's not the quality that's required for generating very, very high quality genomes. What I like to say is every species out there needs as high a quality genome as possible, which is essentially virtually gap-free. And the way we're doing this now is by doing long-read sequencing using PacBio for an example. And in order to do the long-read sequencing, you need very, very high molecular weight DNA to generate the long reads that are required for high-quality genomes. Well, I mean, JGI is a really fantastic place for doing research, and a nice thing that JGI does is essentially a scientist can come up with an idea that falls within the research scope of JGI, and if it's selected, then all of the resources that JGI has can be put on this particular genome or this particular genome project. So it means that the scientist, the one who's thinking about the asking these fantastic questions, they don't have to actually develop their own genome center. They don't have to buy all this expensive equipment. The user facility at JGI, you come up with this great idea, you provide the genetic material to JGI, they give you a fantastic genome, they've annotated it, and then you can go back and ask all the questions you want to ask. So that's a really, it's a fantastic resource, and it's used by scientists around the world.